Hello everyone. Today we are going to talk about protein structure and life cycle. So what is a protein? Protein is a macronutrient that is essential to building muscle mass and it is commonly found in animal products, though is also present in other sources such as nuts and legumes. There are three macronutrients: proteins, fats and carbohydrates, and these macronutrients provide calories or energy. So chemically, protein is composed of amino acids, which are organic compounds made of carbon, hydrogen, nitrogen, oxygen, or sulfur. Amino acids are the building blocks of proteins, and proteins are the building block of muscle mass. So now let's talk about the types of protein structure. The first is called as primary structure, and it describes the unique order in which the amino acids are linked together to form a protein. And proteins are constructed from a set of 20 amino acids. So on your right side, you can see the primary structure of the protein. Now, the second level is secondary structure of protein. It refers to the coiling or folding of a polypeptide chain that gives the protein its three-dimensional shape. There are two types of secondary structures observed in protein. First is alpha helix. This structure resembles a coiled spring and is secured by hydrogen bonding in the polypeptide chain. The other is beta pleated sheet, and this structure appears to be folded or pleated and is held together by hydrogen bonding. The other ones are beta turn, which is between polypeptide units of the folded chain that lie adjacent to one another. And the fourth one is called as random coils. These random coils are mostly seen at the ends of the protein structure, usually at the N or the C terminal of the protein. Now, the third level of the protein structure is called as tertiary structure. It refers to the comprehensive three-dimensional structure of the polypeptide chain of a protein. These are several types of bonds and forces that hold a protein in its tertiary structure. So some of the bonds are hydrophobic interaction, and these interactions contribute to the folding and shaping of a protein. Next is hydrogen bonds, which help in stabilizing protein structure. Third is ionic bonding, which occurs between the positive and the negative charged R groups when the proteins fold. Fourth is disulfide bridges, and they happen because during folding it can result in covalent bonding between the R groups of the cysteine amino acids, and they are also called as disulfide bridges. And the last is van der Waals forces, and these forces assist in the stabilization of protein structure, and these interactions pertain to the attractive and repulsive forces that occur between molecules that become polarized. Now, the last of the level of the protein structure is called as quaternary structure. So it refers to the structure of a protein macromolecule formed by the interactions between multiple polypeptide chain. So now each polypeptide chain is referred to as a subunit, and proteins with quaternary structure may consist of more than one subunit, and these subunits may be same or they may be different. For example, at this level, the protein may be monomer, dimer, trimer, or a tetramer. So this is how we categorize the quaternary structures on the basis of how many subunits they have. Okay, we forgot something. And what is that? That is supersecondary structures. So now let's learn more about them. They are also called as motifs, and it is a folding pattern involving two or more elements of secondary structure. Motif provides function to domain, and they are different types, and simplest among them with specific function consist of two alpha helices joined by a loop region. Can you see the first one? That is called as beta alpha beta unit. The second one on your screen is called as Greek key. The third one on your screen is called as beta meander, and the fourth one on your screen is called as beta barrel. So like I said, they're also called as motifs. So imagine you are a small child and you are playing with your building blocks. So all of us remember building blocks, isn't it? So imagine you're building a tower using building blocks. So from the same building blocks, you can make different types of towers, isn't it? So imagine you have created a protein using these four motifs, and now you are creating a new protein using the same motifs but in a different position, orientation and conformation. So what I'm trying to put forward is that the entire protein structure is actually a big castle which is made by using these small motifs, also called as super secondary structures, which you can imagine as a small building block in the game of Lego. So these small conserved structures are also called as motifs and domains. So now let's talk about the protein life cycle. So what is the life of a protein like? So first of all, a blueprint is transcribed from a gene and eventually becomes translated into a chain of protein building blocks. This chain folds it into itself and into a three-dimensional molecule and then takes on functions such as helping create a cellular structure, forming a molecular machine, or transporting other molecules. The life of a protein ends when it is broken down to its components by a molecular shredder. So, in totality, this is the life of a protein. So now, let's see it diagrammatically. So can you see this DNA which is painted in purple? So this polymerase had just come and was transcribing our purple colored DNA. Let's see it one more time. Yes, so see, the RNA polymerase has come. It is making mRNAs, and now this mRNA is caught up by the ribosomes and the translational machinery, and all the tRNAs are coming one by one and are bringing amino acids with them. And now this is going to make a protein strand. So this is how the life cycle of a protein starts. But do we know how do proteins die? So the level of the proteins within these cells are determined not only by the rates of the synthesis but also by the rates of the degradation. There is a balance in our body, and that balance is called as how many proteins which are still functional in our body and how many proteins which are degraded. One cannot have the 100% fullage criteria of a protein in body, because proteins are in a life cycle and many of them are dying and many of them are being created, and the proteins are being created when they are needed in the body. So the half-lives of the proteins within the cell vary widely from minutes to several days, and differential rates of protein degradation are an important aspect in cell regulation. So when a specific type of signal comes, a specific type of protein set is being created in the body. But when the signal is gone, those proteins are not needed anymore in the body, so they need to go, and that is why the cellular machinery which degrades the proteins is of utmost importance. But because otherwise the accurate and specific cell regulation won't happen. Many rapidly degraded proteins function as regulatory molecules such as transcription factors, and the rapid turnover of these proteins is necessary to allow their levels to change quickly in response to external stimuli. So other proteins are rapidly degraded in response to specific signals, providing another mechanism for regulation of intracellular enzyme activity. In addition, faulty or damaged proteins are recognized and rapidly degraded within the cell, thereby eliminating the consequences of mistakes during the protein synthesis. So in eukaryotic cells, the two major pathways, first the ubiquitin proteasome pathway and the other lysosomal proteolysis mediated protein degradation, keeps on happening in the body. Please stay tuned to watch our next series in protein life cycle. We'll be explaining ubiquitin proteasome pathway and lysosomal proteolysis mediated protein degradation. I hope you enjoyed this video. Thank you. Please like, share and subscribe. I'll see you again.